Hello, everyone. Good afternoon. I'm Dr. Edwin Ma. I'm a hematologist working at the Department of Pathology of Hong Kong Sanatorium and Hospital. So in the next maybe an hour or so, I discuss with you the impact of genomics on healthcare. I wish to focus on the scientific and clinical aspects, but not business development or cost effectiveness, economics, etc. Genomics is a study of genes and their function and related techniques. Right. So while genetics scrutinizes the functioning and composition of single genes, genomics addresses all genes and their interrelationships in order to identify their combined influence on the growth and development of the organism. Right. So back in 2019, I gave a talk at the open day of Hong Kong U Space. And at that time, the talk title was The Future of Genomics in Laboratory Medicine. And in 2019, I described molecular pathology as a new discipline. Molecular pathology is an emerging discipline within pathology, which is focused in the study of disease through examination of molecules within cells. The purpose is to elucidate the mechanisms of disease by identifying molecular and pathway alterations. And at the core of this discipline is the application of classical and novel techniques developed in biochemistry, cell biology, molecular biology, proteomics, and genetics to the evaluation of pathological processes. And molecular pathology now basically use a lot of gene sequencing techniques. And back in 2019 at the lecture, I described genomic sequencing, the transition from Sanger sequencing, so-called first-generation sequencing, to next generation or second-generation sequencing. And in this talk, we shall look at further advances since that time. So back then, I described the important clinical applications of genomics, mainly to define the disease and to diagnose disease, to predict the patient outcome. For the doctor, the fourth point is most important, to find the drug target, to monitor treatment response after starting appropriate treatment, to look for any genetic susceptibility factors when there's a positive family history, and then last but not least, to screen disease. Okay. So this is the last slide back in 2019. Okay, so at that time, I put forward some future perspectives. Okay. First is pushing the technical boundary. Second is expanding the scope. And then making the technology more affordable, and then try to identify gaps and to fill these gaps of the genomics approach. Okay, so in this talk, I shall lead you through what we have done since 2019. Okay, so in these 3 years, what have we done? Okay, so first of all, the technical boundary. Okay, so how are we doing in 2022? So 3 years down the road, what is the development in these areas like long read length, NGS, and also minimal residual disease testing? In fact, I'm pleased to let you know that in fact, we are making very good progress. We have done a lot, and I shall highlight it in this talk. Okay, so first of all, the long read length NGS. Okay, so this figure, okay, shows the transition from the first generation Sanger sequencing, okay, to next generation sequencing. And in fact, there are now several different, different transition through different models of NGS from the pyro sequencing based 454 sequencing, okay, which is now being phased out, okay, to, for example, Illumina based sequencing, right? And then there is now development of the third generation NGS, okay, which is the Oxford Nanopore, okay. The read length is very important, okay. So while Sanger sequencing, we are talking about 1,000 base pair read length. In the Illumina, like a MySeq sequencer, we're talking about a short read length, okay, typically 300 or even shorter, okay, like 150. Okay. And for the third generation NGS, nanopore sequencing, we are talking about a long read length, okay? Like, for example, over 36 kb or even longer, okay? So what is the advantage of a long read length over short read length? And why do we need to have a long read length NGS? In fact, the reason is mainly for the detection of structural variation, okay? It's quite challenging to detect this type of genetic variation by a short read length NGS, okay? But first of all, we step back a little bit and describe what is a structural variation, okay? So structural variation is generally defined as sequence variants involving a DNA region of approximately 1 kilobase, 1,000 base pairs, or even larger, but can also be operationally defined as anything bigger than 50 base pair, okay? So the scope, in fact, includes large deletions, duplications, inversions, insertions, and chromosomal translocations. But note that the deletions and duplications or insertions can also be termed as copy number variants. And broadly speaking, I mean, for the broader definition of structural variation will also include the copy number variants, okay? But sometimes structural variation only refers to the chromosomal level translocations or inversions. But the bottom line is these are large-scale genetic changes. Okay, so in this figure, you see two normal chromosomes, okay, with the sequence labeled in blocks, okay. So in the first chromosome, the normal chromosome, suppose, okay, the sequence is arranged A, B, C, D, E, okay, and in the second chromosome is V, W, X, Y, Z. So for the inversion, okay, using the first chromosome example, inversion would be A, D, C, B, E, okay. A deletion would be A, B, C, okay, and then E. The duplication would be, an example is A, B, C, 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 D, E. And then finally, The translocation would be exchange of chromosomal material, okay? For example, the VW in red of the second chromosome is fused to the CDE of the first chromosome. But because this is the exchange, okay, between two chromosomes, so you also have the reciprocal fusion, okay, the AB with X, Y, Z, okay, on two different chromosomes. Okay, so in fact, what are the problems of short read length NGS in the detection of structural variants? Mind you, the short read length NGS reliably identifies single nucleotide variants and the small insertions and deletions, okay? So SNV and the small insertion deletions are not a problem. But when applied to the structural variant detection, this approach cannot completely overcome the limited sequence and the insert size of the standard short read length NGS. Because notably, the short read length fails to identify most large insertions, deletions, and CNVs in repetitive regions of the genome. Since these structural variant breakpoints cannot be captured directly from single read alignment when they are embedded within or composed of repeat elements that are confound mapping. Okay, so the keyword is repeat elements will be very challenging for short read length. For example, if we are talking about chromosomal translocation, then this non-allelic homologous recombination of repetitive sequences is thought to be a predominant mechanism. Okay, but however, this non-unique sequence franking this structural variations are often not detectable by sequencing-based methods. Again, if you step back a little bit, it's the hot topic. In fact, the methods of detecting structural variation are as follows, okay? The low-pass whole genome sequencing, whole genome sequencing, long read length sequencing, namely the Oxford Nanopore and the Pacific Biosystems, mate pair sequencing, RNA-seq fusion panel. Okay, so this look for the chimeric fusion transcript, and then finally, the new technology, optical genome mapping. Okay, so these are, in fact, very hot topics for clinical studies and also investigation. Okay, so we zoom in and see what we are doing with the long read length NGS, especially with the Oxford Nanopore. Okay, so this is the mean iron. There are other more scaled up versions, but this is the like a palm size cartridge, okay, in which you've got your flow cell here, and then the machine, and then there's a connection to the computer. Okay, so with this nanopore sequencing, so what we are talking about is that with your genomic DNA, okay, after the addition of the suitable adapters, okay, so they are loaded into the flow cell, and in fact, will generate a so-called real-time sequencing. That means the sequence are read real time. Okay, so you no need to complete the experiments before you read the sequence. Okay, and at a speed of 450 base pair per second. So this is the nanopore. Okay, and you see the DNA sequence going through it. Okay, for the actual sequence to be read. And in fact, you remember we term it as a third generation NGS, but people are also referring it to a third generation cytogenetic analysis because of its capability to detect chromosomal level abnormalities. So it's equivalent to a cytogenetics, but you are not doing a metaphase analysis. Instead, you are using an NGS approach to do cytogenetics. So it's termed third generation cytogenetics, diagnostic application of long read sequencing. In fact, we are also doing that in the lab. So just to illustrate with an example, okay, so we are talking about a Philadelphia chromosome positive acute lymphoblastic leukemia. So this is a type of leukemia driven by a chromosomal translocation, okay, a balanced translocation between chromosomes 9 and 22, okay, termed the Philadelphia chromosome. And this is a 66-year-old lady with a B-cell acute lymphoblastic leukemia. And when we do the RT-PCR, okay, with the BCR-ABL fusion transcript, okay, you can see that the patient is positive for the 190 BCR-ABL fusion transcript, okay, but negative for the P210, okay. And this is BRCA2 control for the RNA integrity, okay. So RNA is good quality, and the patient is positive for the P190 BCR-ABL, so Philadelphia positive. Okay, so this is very straightforward, okay. You have got the correct diagnosis. And so a week later, we got the chromosome results back. And in fact, we do find the Philadelphia translocation, okay. So this is the elongated chromosome 9, so-called derivative 9. And this is the shortened chromosome 22, okay. So this is the Philadelphia chromosome, a shortened chromosome 22 or derivative 22. But in fact, this patient also has other abnormalities like monosomy 15, monosomy 20, and there's also something wrong here with the chromosome 6 and also chromosome 3, okay. But the main driver is this Philadelphia translocation. So the question is, can we use an NGS? So instead of actually looking at the metaphase, can we use an NGS to pick up the translocation? And so this is also a structural variation, okay, a chromosomal level change. And in fact, the answer is yes. Okay, so if you look at the schematic diagram of the Philadelphia chromosome, so we are talking about an elongated chromosome 9, a shortened chromosome 22, okay, in which it carries the 5' BCR-ABL, okay, 5' BCR on a 22 and a 3' ABL, okay, whereas on the derivative chromosome 9, the elongated one, you see the reciprocal fusion, okay, the 5' ABL, chromosome 9, and the 3' BCR, chromosome 22, okay, so remember this. And for the P190 transcript, okay, so we expect the B-cell breakpoint to be at exon 1, okay, here. Okay, small m or minor B-cell breakpoint, okay? Because the patient is negative for P210, okay? So we do not expect the breakpoint at the major or the capital M BCR, okay? So we expect a breakpoint exon 1 of the BCR joining to the ABL, okay? So the question is, can we use the long read length Oxford nanopore sequencing to pick it up? And in fact, the answer is yes. This is a bit busy, but then, so I lead you through it. So this is chromosome 22. Okay, so the breakpoint is at 22q11. And this is the chromosome 9, and the breakpoint is more distal, 9q34, okay? So at the bottom, this is a normal control. So you see all these reads mapping very nicely to 22 and chromosome 9, okay, with no breakage. But, okay, on the top panel for our patient, you can see a lot of this breakage, okay? And in fact, they can align. For example, we're talking about, you remember, so the BCR-ABL is 5 prime, 22, and then 3 prime chromosome 9, okay? But can we pick that up? And in fact, the first read is derivative 22 forward, okay? And then that maps to the chromosome 22. So this is the 5 prime 22. But where's the 3 prime? So the 3 prime is actually nicely mapped to chromosome 9. Okay, so this read. Okay, so on and so forth. So I think in summary, we can very nicely, by the log-linked NGS, okay, we can actually map the Philadelphia translocation. Okay, so the forward derivative 9 is supported by 3 reads. The reverse derivative 9 by 1 read. Forward 22, 2 reads. Okay, so this is very important. This is the 5 prime BCL, 3 prime ABL, and then the derivative 22 reverse supported by 4 reads. Okay, this is actually the reciprocal fusion, and then 8 wild type reads. Okay, because there's mixture of leukemic and normal cells, and total 18 reads. So then with the long read link NGS, we can very nicely map the Philadelphia translocation. Okay, how about the other aspect? Okay, technically, okay, can we push the boundary in minimal residual disease testing? Right. So what is minimal residual disease? Or more correctly, it should be termed measurable residual disease. But both are abbreviated as MRD in short. So this refers to the small number of cancer cells that remain in the body after treatment. So the number of remaining cells may be so small, they are typically very small, okay? And they escape detection by your clinical examination and also escape microscopy, okay? So molecular method is the only way that you can confidently pick up this small clone, okay, MRD. It's like the tip of the iceberg. So I think morphology can only pick up the iceberg that is above the water, okay? In fact, it's only a minority of the whole iceberg, okay? So that means what is beneath the water, you have to use more sensitive MRD methods to detect. And for this iceberg, if you translate it into the disease, okay, it's like that, okay? So you see the iceberg, what is above the water is this red line, okay? Red line is the detection limit of morphology, okay? So the y-axis is the MRD level. So here we are talking about the 5%, okay? 5% level. So beneath the water, okay, first of all, you have the quantifiable MRD, okay? So it's down to now typically 10 to the -5 level. That means quantifiable MRD, usually the limit is 10 to the power -5, that equivalent to 10,000, one tumor cell out of 10,000 cells, okay? It's very similar to chemistry where you have the limit of quantification, okay? But beneath 10 to the power -5, okay, you still have MRD, but it's non-quantifiable, okay? Just like if you're below the limit of quantification of chemistry. And then finally, down further, you have got non-detectable, okay? Again, similar to the limit of detection, okay? So just remember now for the current methods, the limit of detection would be in the 10 to the -5 or at most 10 to the power -6, okay? That means one cancer cell out of 10,000 to 1 million, okay? That's the limit of MRD detection, okay? But also remember, below that, we're talking about is not the MRD total negative for leukemic cell. It's only that you cannot detect it, okay? Because it's lower than the limit of detection of current methods. Okay, so the clinical impact, okay, because this talk is on the impact of genomics, so the impact would be, of MRD study is to provide a more quantitative assessment of the deep remission. Okay. That means, say, like for 2 patients, if both are in complete remission, okay, we do not know which one has an MRD. Okay, so therefore, in one patient could be complete remission, but MRD still positive, but the other patient also in complete remission, but it's MRD negative. Okay, so it actually means a lot to these patients. Okay, and also to define, second is to refine post-remission relapse risk assessment and prognostication. Okay, because MRD positive patients are more prone to disease relapse, whereas MRD negative patients are less prone to disease relapse. And if you are able to predict relapse, then you can identify impending relapse, like an increasing MRD level, to enable early intervention. And finally, MRD can serve as a surrogate endpoint. That means MRD negative can be a surrogate for survival in clinical trials. Because nowadays drugs are very effective, and for survival analysis, you need a very long follow-up period. Whereas for MRD, okay, you can more quickly reach the endpoint, and then you, so as to accelerate the drug testing and also approval. Okay, so currently at our lab, we are doing a lot of MRD studies by various techniques. Okay, but one technique I wish to discuss with you this afternoon is the digital droplet PCR, okay, or ddPCR in short. Okay, so we use ddPCR for AML, acute myeloid leukemia MRD studies, and also for in some difficult CML patients with variant BCR breakpoints. Okay. So ddPCR enables monitoring of deep MRD, okay, because it has a very good analytical sensitivity for small clones. Okay, so what is ddPCR? Okay, so in fact, I think the gist is that here you actually partition out the DNA molecules. Okay, for example, with a water oil immersion droplet. Okay, so we are using droplets to partition out the DNA molecules, for example, into 20,000 droplets, and then PCR amplification of each of molecules inside the droplets. Okay, so actually massive sample partitioning is the key. So what is the advantage? I think you can imagine if you have a group of 1,000 people, and if you want to identify 1 people out of the 1,000 by PCR, it's not that easy because there's a lot of primary competition and also other interfering factors. But however, if you try to separate this 1,000 people, and then you try to separate each person into one room, and then you look for the person in each room, then there's a much higher chance for you to identify the person. Okay, so in fact, the aim is to either have 1 target or 0 target into each of these droplets. Okay, so it's either 0 or 1. Okay, that means it's digital 0, 1, just like a computer. Okay, so that's why it's called droplet, because droplet to separate out the molecules, and then digital is to aim for either 0 or 1. But then life is not so simple. Okay, you're bound to have more than 1 copy, right? Especially if the MRD level is high. Okay, but in fact, you can use actually a arithmetic formula, so-called the Poisson logistics, in order to actually calculate or derive the copy number, actual copy number in your sample. So the advantages over real-time PCR is that there's this is absolute quantification. of copy number. So you no need for you to do the standard curve, okay? And then you have superior precision at low levels. And this is a relatively simple and robust procedure because this is an endpoint PCR, okay? It's not it's an absolute quantification and endpoint PCR and not a relative quantification. Okay, so just to draw an example, okay, we use ddPCR to monitor NPM1 mutations, okay? And we are doing an RNA-based assay, okay? So they are reverse transcribed to cDNA. And then here, the NPM1 mutation are clustered exon 12, okay? So basically, what we are doing is we use a common forward primer at exon 11, and then we designed mutation-specific reverse primers, okay? And then the probe is labeled with, say, like a FAM fluorochrome, okay? So this is target detection, and we always normalize with a control, okay? For the internal control, we use the ABL1, okay? So what we are reporting is the copy number of the NPM1 over the ABL, okay? So normalized and then as a percentage ratio, okay? We look at how useful it is using two patient examples. So this one is NPM1 non-type A, okay? So the insert is CTTG, whereas type A, the more typical one is CTCG, okay? And then the y-axis shows the normalized ratio, okay? Is this NPM1, okay, over the ABL, okay, as a ratio. So you can see this patient, in fact, after treatment, still MRD positive, okay, at a low level. This is 0.96%, okay? And then rapidly dropping to undetectable. And then this is a persistent MRD negative, okay, over nearly a year, okay? So this is very good, okay? The patient is, this one is complete remission and then MRD negative. And the patient did remain in complete remission, okay? So the MRD negative is predictive of a long-term remission. But for another patient, okay, so this is a similar type of leukemia, okay, just like the previous patient, okay, also NPM1, but this time it's a typical type A, and also a FLT3 IDD positive, acute myeloid leukemia. So this patient is less fortunate, okay, so despite receiving a bone marrow transplantation, the patient still remains as MRD positive, okay, so this should be 51%, okay? And then, because still MRD positive, the patient was given this FLT3 inhibitor, sorafenib, and managed to drop the MRD, okay? But still positive, so the patient received the donor lymphocytes, okay, from the BMT donor, okay, and managed to drop the MRD furthermore. But then the patient suffered side effects of the treatment, okay? So both treatments were withheld, and then the MRD starts to creep up, okay? So the doctor decides to change the treatment, okay? Add in the hypomethylation agent and a BCL2 inhibitor, okay? And then manage to drop the MRD to undetectable for around half a year. Okay, but then it creeps up again and has to resume treatment, okay, resume the lymphocyte infusion from a donor, but then this time doesn't work, okay, and the MRD actually shot up to very high levels, and then the patient relapsed and then succumbed to the disease. But the message is that the MRD is very useful to actually guide the treatment because the doctor actually really base the treatment upon the MRD result. Okay, but mind you, in clinical practice, although MRD is very useful, but always remember a single mutation does not provide the full picture, because we do encounter escape mutants. Like, for example, even if the patient is NPM1 negative, okay, it may still relapse because of an NPM1 independent clone. And then always compare the normalized ratio with that in the diagnostic sample, okay, for proper interpretation for the so-called log reduction. And then always remember monitoring by RNA may show slightly discrepant result with the DNA. Okay, so always mindful of the starting sample in order to interpret the MRD result better. Okay, so I think in these 3 years, we are doing quite well in pushing the technical boundary. So how about expanding scope? Okay, so you remember when I gave the talk in 2019, okay, we still did not have COVID at that time. I remember it's November 2019, okay? So at that time, we did not have COVID, okay? And when we have COVID, in fact, we see a lot of application of genomic study in in helping to tackle and also to try to combat COVID. I think one genomic method that stands out pretty well would be again the sequencing. Okay, so we also did genomic sequencing of the SARS-CoV-2. In fact, the government and also university labs are also doing that. Okay, so in fact, what are the important reasons why we do sequencing of the virus? It's because, first of all, is to monitor the trends at the national level. Okay, and then better understand epidemiology at the local level. I will illustrate with examples. Okay, like for example, in a hospital outbreak or in a community outbreak, like in the elderly homes or schools, okay, or other settings. And there are also other clinical utilities, okay, like identify the sources of infection, identify super spreaders, and to predict the severity by actually knowing which variant is involved. Okay, so the SARS-CoV-2 is an RNA virus. Okay, the genome okay, shows 30,000 nucleotides. Okay, so not too difficult to sequence. And in fact, note that the mutations in the genome produces a fingerprint, or like the ID card of the virus, of that viral strain, that can be used to infer ancestral relationship or so-called phylogeny. Okay, so that's the reason, okay, why we do genomic sequencing. Okay, it's just like to find the ID card, okay, from the wallet of that person in order to identify the person. Okay, so the method we use is a direct whole genome sequencing. Okay, so we sequence direct from a clinical specimen. Okay, like the nasopharyngeal aspirate or from the deep throat saliva, because this will minimize the mutations introduced from viral propagation or passage. Okay, and okay, just like previous example, we also use the nanopore sequencing. Okay, and because the wild genome is only 30 kb, okay, so we can have a very quick result. Okay, usually available on the next day, sometimes even on the same day. Okay, so I just draw on our early experience of 26 cases and just illustrate with how we investigate a hospital outbreak. Okay, so for the wet lab, we extract the RNA directly, okay, from the specimen and then do a reverse transcription, okay, do a whole viral sequencing, okay, on the Oxford nanopore mean iron, okay, just like in the leukemia case. And for the dry lab, we use this Arctic bioinformatics protocol, okay, and then do a phylogenetics using the next clade. Okay, I've shown you this before. So this is the palm size sequencer, okay, and we have a number of these, okay, all hooked up to the computer. Okay, so in fact, in November 2020, okay, we are hit by the fourth wave. So you remember this is the dancing and singing group, okay, and then we've got two hospital clusters, okay, one in the dialysis center and then the other in a ward, okay. So in the dialysis center, we've got a 72-year-old man who required dialysis because of diabetic nephropathy, okay, and he was on hemodialysis, okay, but developed a cough and sore throat. And then the saliva was positive, okay? So this is the man in the dialysis center. And then we also had another patient at roughly the similar time, a 77-year-old woman with colon cancer who developed fever and then has a heart rhythm problem and then chest X-ray infiltrates, okay? And rapidly deteriorated and actually died a day later, okay, after symptom onset, okay. And then she was also tested positive. Okay, so when we look at these two patients, okay, so this is the epidemiological investigation, okay. So first of all, for the Ward A, okay, so this is the index patient. Okay, this is the patient with the colon cancer. She was tested positive here at 26, okay, and she had symptom onset on the 25th, okay. But in fact, if you look at her visitors, in fact, she was visited by a daughter, okay, or 3 days beforehand, and the daughter was also symptomatic, okay. And this one belongs to the singing and dancing cluster. Okay, so in fact, there was also later on, in fact, there's a ward attendant, okay, also infected, and then subsequently 3 nurses also infected, okay. So this is situation for the ward, okay. In the dialysis center, okay, the patient, okay, who had diabetic nephropathy on hemodialysis, okay, had symptom onset, okay, here, okay. But then, in fact, if you look at it, his wife, okay, had symptom onset 2 days earlier, okay. And then this particular patient also spread the virus, okay, to another patient, okay, in a nearby bed, okay, in the dialysis center, okay. So the purple color refer to the exposure, and then the yellow is the symptom onset, okay, and then the red one, this colon cancer patient actually passed away 1 day after confirmed to have COVID. Okay, so the question is, could we confirm that in fact they have a strain identity, okay, and what is the relationship of these viruses, okay? So we did the whole genome sequencing, and this is again quite a busy slide. Okay, so in fact, the first 3 patients were from a dialysis center. Also, I think the bottom, and then the rest will be from the ward. So I think the bottom line is, okay, these all viruses, okay, they share a lot of genomic sequences, okay. So they belong to the same strain, okay. But in fact, if you look at the renal dialysis center, okay, although they show the same strain, okay, but they have a different identity because the renal dialysis center, okay, all these 3 patients share the same SNP that is not found in the ward, okay. So in fact, it is not the, so there are actually 2 separate introductions, okay. It's not that the patients spread from the dialysis center to the ward or vice versa. And secondly, in fact, the other patient from the dialysis center again show a separate SNP. So there is evidence of viral evolution in the dialysis center. So that's the first point. The second point is in the ward, okay, although they belong to the same strain, in fact, there's also evidence of viral evolution because the 2 separate samples from the son of the patient and also a sample from a nurse, okay, show a distinct SNP, okay. So this SNP shows that in fact, although the virus come from the same source, in fact, there's evidence of evolution. So if you do the phylogenetics on the next clay, okay, they actually belong, okay, to the same clay, okay, but with some minor alterations as I've illustrated earlier. Okay, so finally, have we closed any gaps of a genomics approach? Okay, and what would be the current situation in terms of application of genomics? So again, in these 3 years, comprehensive genomic profiling now becomes standard practice, okay, both in private and also public sector. Okay, so for example, we now currently are able to do our own comprehensive genomic profiling on this relatively large gene panel, okay, and then covers all the spectrum of genetic variants. And to close the gap of the genomics approach, we have been doing cell therapy, advanced cellular products like CAR T cells, chimeric antigen receptor T cells. Okay, so we have been doing that. Okay. But, however, there are still, okay, some issues. It's not only in Hong Kong, but internationally, because there's just a recent publication just last week from the New England Journal. The title is Closing the Gap in Cancer Genome Testing. Okay, so what are the gaps that are still remaining? And in fact, the gaps from the paper refers to these points, okay? So which biomarker to order, okay? So the doctors would need advice as to which biomarker to choose. And then turnaround time, okay? You need to have a quicker result, okay? And then interpreting complex genomic test results, okay, is challenging because the multiple genomic knowledge base are available, but not harmonized with respect to content format, okay, or evidentiary standards for actionability, and oncologists may not know which one to use. So here, the pathologists or the lab, okay, or the scientists have a lot of role to play, okay, as to advising which marker to order, and to help to interpret it. So I think in our hospital, we are very fortunate to be able to run a molecular tumor board with Hong Kong U Department of Oncology. Okay, in that we receive referrals from doctors on how to interpret the complex genomic results. Okay, and then we have in this tumor board, we have pathologists, we have scientists, geneticists, bioinformaticians, all of these experts, okay, helping to, okay, interpret the genetic variants and to advise on cancer treatment, especially whether there are actually target drug available, either FDA approved or off-label, or whether still in clinical trials. Okay, any need for further tests, like whether you need the germline test, or you need any orthogonal validation of the genetic results, any monitoring recommendations, etc. Okay, so I think we at least it's a good start. Okay, we try to tackle the issue and then try to close the gaps that are being brought up by the paper. But that's not all, because there are also other gaps that is beyond our scope, okay? Because this is basically for the oncologists to tackle, because it can be unclear what level of evidence support the selection of potential treatment. That means whether the level of evidence is strong enough for the doctor to use a particular treatment. Oncologists may not know how to obtain access to the treatment if it is investigational or off-label. And then there's also incomplete and variable collection of data on patient outcome, okay, which complicates data sharing among practices and health systems, and it hinders ongoing evidence generation. Okay, so this is for the oncologists to tackle. Okay, so I think in summary, in these 3 years from 2019 to now, we've been doing very well, and we're actively applying genomics to healthcare. And I think in the near future, what we are hoping for is to actually use more whole genome sequencing, okay, for clinical application, okay, not for research, okay. So that's in the near future. We hope to do more whole genome sequencing, both in constitutional disorders and for cancer purposes. And then one thing is that we are also very eager to see the results that are being generated from the Hong Kong Genome Project, okay, the Hong Kong Genome Institute, okay. I'm sure that they will bring new light into the application of genomics to healthcare. So I think with that, I close the talk, and then thank you very much for the attention.